Okay, so thank you very much. Good evening to everyone. Thanks, Neil, for the introduction, and thank you for the invitation. It's a pleasure to be here. Congratulations on the project and on the achievements during these years. I was saying before you go for a second round, I would love to get involved and contribute somehow. So congratulations. And I'm going to talk about this protein here, LSECtin, which is an innate receptor, and the role that it has in the regulation of inflammation during advanced liver disease. These are conflicts of interest, and let's just start in the beginning. So, as you all know, the hepatic immune responses or activity is characterized by this, let's say, tolerogenic state, which is developed in response to the constant exposure to low amounts of harmless antigens. And in response to this situation, the liver brings the induction of anti-inflammatory cytokines such as IL-10 or TGF-beta, and also induce the expansion, the proliferation of regulatory T cells that you probably know that are hot topic again because of the Nobel Prize, the recent Nobel Prize. So through these two mechanisms provides a tolerogenic state during homeostasis. However, this condition can change, and when an antigen reaches the liver, a pathogenic antigen reaches the liver, or there's some kind of threat, this tolerogenic state changes towards a local immune activation that it's supported by the recruitment of cells from circulation, neutrophils, there was lymphocytes, monocytes, and then they provide what we call a physiological inflammation, which is going to be responsible not only for eliminating the threat, but also for restoring homeostasis afterwards. And this is very important. Okay, so this balance can be dysregulated, of course, and for instance, if we have an excess of tolerogenic response, we can have problems such as progression of tumors, or we can have viral infections. And on the other side of the coin, as it happens in cirrhosis, if we lose mechanisms of tolerogenic response, then we can get chronic inflammation. And chronic inflammation comes with increased hepatic cell death, with the initiation of fibrogenesis, and these processes may lead to organ dysfunction. So it is important to understand how local immune response works and how these tolerogenic mechanisms are lost in cirrhosis. Okay, so the liver is considered from our side as a very important organ in immune surveillance. In these activities provided not only through all these recruited cells that I've just mentioned, the neutrophils, the monocytes, and lymphocytes, but mainly due to the number of resident immune cells that are here listed in the left side of the slide, and among which there are two very important populations, Kupffer cells and also liver sinusoidal endothelial cells. In fact, these LSECs are located just between the liver and circulation. So just because of this, they are going to constitute like a physical barrier separating the liver from circulation, and thus also like a first line of defense against all antigens or pathogenic antigens reaching the organ. So these cells are going to be very important in inducing and above all in maintaining or keeping this tolerogenic state during homeostasis. The immunological functions of LSECs go beyond this physical barrier, and for instance they have very high phagocytic capacity because they express a lot of innate receptors such as TLRs, lectins, FC receptors, and all these receptors can recognize antigens and internalize them, so they have high phagocytic activity. But they also are important in leukocyte recruitment. For instance, they express CXCL10, which is important to attract leukocytes, and also adhesion molecules such as VCAM, which is going to be important for extravasation of leukocytes into tissues. But the main immune modulatory activity of LSECs is provided by this second line here through the expression of TGF-beta, and also because they have the ability to present antigen either without co-stimulation, which leads to the expansion of regulatory T cells, or with inhibitory ligands such as PD-L1. Another mechanisms for LSECs to provide this tolerogenic stage is the expression of LSECtin, and this is a gene, CLEC4G. It's a group 2C type lectin, which belongs to this small family over here, which is expressed in hepatic antigen-presenting cells. As a presenting cell, of course, it's able to induce phagocytosis, and it also contributes in modulating signals provided by other innate receptors, such as TLRs. And in fact, LSECtin can negatively regulate T cell responses in acute liver injury. And this was demonstrated several years ago working with an LSECtin knockout model of acute hepatitis, in which authors showed that there was a significant increase in transaminase levels compared to the wild type model, and also at the same time there was a significant increase in the amount of cells TNF-alpha positive, interferon gamma positive cells compared to wild type injured mice. This was in acute liver injury several years ago, 2009, but years later, the gene expression levels of several innate receptors were tested in two models of chronic liver disease, in this case in rats, both in CCL4 and in bile ligation, in BDL, and many of them were dysregulated, and here appeared LSECtin as the only one that was repressed during the cirrhosis protocol, in both protocols, both in BDL and CCL4. And of interest, if we had these LSECs from these rats and put it in co-culture with T cells, there was an expansion and proliferation of I-17 positive effector T cells, suggesting that this repression in LSECtin was inversely related with the increased pro-inflammatory expansion in the liver of cirrhotic rats. This was chronic liver disease, and we had two different evidences on the importance of LSECtin in liver damage. LSECtin has different ligands. These are CD44, BTN383, and here LAG3, which is the only one that has been demonstrated to inhibit the T cell activation through an early interaction with a TCR, which provokes the inhibition of effector cells, the promotion of regulatory T cells, and this is the most public candidate to study the possible mechanism of tolerance for LSECtin and LSECs. In other settings, for instance, the interaction between LSECtin and LAG3 and this ligand has been related with the progression of melanoma, but very little is known about this interaction in advanced chronic liver disease. So in the last few years, we've been very interested in understanding how this loss of LSECtin can modulate the progression of pro-inflammatory TH points in cirrhosis, and maybe trying to consider whether retreating LSECtin is able to reduce the hepatic tissue damage. So the first thing we were interested in was trying to know whether there was any kind of translational interest or value in the loss of LSECtin in cirrhotic patients. And for doing that, we work with two different cohorts of patients, one from our hospital in Alicante, another one from Clinic de Barcelona, and also from a series of healthy controls. And we got some liver samples for different experiments that I'm going to show you. So of course, patients have a progressive increase in the deposition of collagen, and they also had from compensated to decompensated stages of the disease. They also have an increased infiltration of macrophages in the liver, shown here by the marker TIM4. Nothing new so far, but very interestingly, these same patients had the opposite behavior for LSECtin. We have like a very high expression of LSECtin in the healthy subjects, and then progressive downregulation of LSECtin in patients from compensated to decompensated stages. And this evidence that we found histologically, they were confirmed both by Western blot and also by gene expression levels of gene of 24G. So it was quite nice, it was quite progressive, and this was some translational value for the study of LSECtin in cirrhosis. We also wanted to check whether this loss of expression of LSECtin in cirrhotic patients was correlated with an expansion of pro-inflammatory Th responses in the liver. So we studied different transcription factors that trigger the differentiation of the different Th responses. For instance, Tbet for Th1, Rho gamma T for Th17, and also FOXP3 for regulatory T cells. And we observed that there was like a progressive increase in the expression of Rho gamma T positive cells in the liver of zygotic patients going from compensated to the compensated stages. So again, we have this this inverse correlation between the increase of LSECtin, and in this case the decrease of LSECtin, and the increase of inflammation in the liver. And for evaluating again this inverse relationship between the two genes, we got to a third independent cohort of patients that had publicly available transcriptional data, and we observed that this was confirmed also in this cohort of patients, and that there was like an inverse correlation between these two genes in zygotic patients. So very interesting for us, and it has translational value. It's great. Very interesting. We also wanted to know what cell types are expressing LSECtin in cirrhotic livers and in mutations. We did this by immunofluorescence, and you can see here C31, which is a marker for LSECs, TIM4, marker for macrophages, LSECtin in blue. So everything you see in purple is the co-localization of LSECtin in LSECs, which are the main cell type expressing LSECtin in human cirrhotic livers. We validated with a second marker, C36, but I don't have it here in this image, and I forgot to clarify about it. So but it was quite clear that LSECs were the main cell type responsible for LSECtin expression in the liver. So we went back to the lab. We were interested in understanding how LSECtin worked in terms of signaling, also what LSECtin-induced cytokine production was, and also to confirm whether this LSECtin could interact with other innate receptors such as TLRs. So for doing that, we worked with two different immortalized LSEC cell lines. One of them we used it as control, and the second one we partially repressed LSECtin using CRISPR tools. And okay, so first thing we did was to confirm that we were able to repress LSECtin in our cell line, and this was shown here. We had around 50% of decrease in LSECtin expression in the CRISPR cell line. And with these results, we culture the cells with the ligands of LSECtin. It's the ligands that I told you before. Here you have CD44 and LAG3. And you can see here how the control cell line increased. Well, we tested different signaling pathways. You have in here AKT, NF-kappa B, and P38. I'm only showing you P38 because all of them behave similarly. And as you can see here, the addition of ligands for LSECtin induced the expression of P38 with both ligands, both CD44 and LAG3. And in the CRISPR cell line, this was again inhibited, confirming that we had a good model for studying repression of LSECtin. Okay, so after doing this, we wanted to know what was the secreton that these cells were able to produce. So you have here the secreton both for ligands, and what you can see here how pro-inflammatory cytokines such as TNF-alpha or I1 beta were significantly upregulated in the CRISPR cell line, suggesting that we have less LSECtin expression. Again, we have a pro-inflammatory expansion in the secretome. And the opposite, cytokine such as IL-10, which was also reversed here, it's right here, behave the other way around. When we have the CRISPR cell line, cytokines such as PI10 were downregulated. So the secretome was directly influenced by LSECtin expression and its ligands. And the third thing that we wanted to know about this innate receptor was whether it would interact with other innate receptors. We have many of them in an antigen-presenting cell. So this is very important because nothing in nature works by its own, at least in immunology. So for doing that, we tried two different things. We culture cells with ligands for TLRs, for TLR2, 4, and 9, either alone, as here, or combined with ligands for LSECtin. And we did the same as before. We studied whether there was a difference in expression of the different signaling pathways. I'm showing you just P38. And if we just represent this combination as the difference in expression related to the signal of the TLR alone, you can see that they behave completely different for the control cell line or for the CRISPR cell line. So this confirms that LSECtin can modulate the signaling provided by TLRs very clearly. Whether it reduces inflammation or not, for that we need to again go to the secretome, and the secretome can be seen here as before. We're seeing here to TNF-alpha and IL-6, and the induction of these cytokines produced by the TLR ligands is significantly downregulated when the ligands of LSECtin are in the culture, and then significantly upregulated again in CRISPR cell lines. So altogether, it's like very focused in vitro evidence of LSECtin modulation of TLR signaling. What is expected somehow, but you have to see it. That's what LSECs through LSECtin can produce. We are also interested in knowing what things can induce LSECtin. Okay, so we tested both pro-inflammatory and anti-inflammatory cytokines. Here we're looking at a bunch of them. These are pro-inflammatory, these are anti-inflammatory, and the gene expression levels of CLEC4G change very clearly. TNF-alpha and a combination of several pro-inflammatory cytokines downregulate CLEC4G. On the other side, anti-inflammatory cytokines, many of them can significantly upregulate LSECtin in this cell line. Okay, so this was in vitro. We also wanted to see some evidence in vivo, so we moved to this CCL4 model of cirrhosis induced by CCL4 orally for 12 weeks, and we were interested in doing this because it was the only way to get primary LSECs and see how they work in culture. So, of course, what we tested first, that we had collagen deposition and infiltration of macrophages and so on. And we also tested whether LSECtin was expressed in the model. So, as we saw for human, for liver samples before, there looks like a very nice expression of LSECtin in the sinusoids in the control mice, and then loss of expression of LSECtin in cirrhosis, confirmed by Western and also by gene expression levels. So behavior was similar to what we had seen before. So we could use these primary LSECs that were defined, were sorted cells defined as CD45 negative, CD31 positive, and CD146 positive cells. So we collected these primary cell lines by cell sorting, and of course we confirmed that LSECtin was downregulated in them. And wanted to know how to stimulate them with anti-inflammatory cytokines to see whether they behave similarly to our cell line. And as you can see here, again, anti-inflammatory cytokines, IL-13, and combination of several of them significantly increased CLEC4G in primary sex. So this first part, just to show you the graphical outcome of the of the paper. So we have like several different evidences. The first thing is that the ligands from LSECtin can induce intracellular signaling in LSECs, and they can do it by their own. By more importantly, they interact with signaling of TLRs, and the anti-inflammatory signal that LSECtin produces modulates the pro-inflammatory signals that TLRs use. This doesn't mean that it cannot interact with other innate receptors. Probably it does it as well. And they do it through the many different pathways that we have tested. All of them have shown differentiation. And second line, it's that anti-inflammatory cytokines can induce LSECtin, that pro-inflammatory cytokines inhibit LSECtin, and the most important part is that in vivo, not only in the animal model but also in cirrhotic patients, LSECtin is very downregulated in the liver, and is inversely correlated with the Th17 response characteristic in the liver of cirrhotic patients. So is where a lot of evidence pointing to the fact that LSECtin is providing the loss of LSECtin is providing some kind of mismodulation of the homeostasis during cirrhosis. So with this, of course, we did all the mechanistic stuff and so on, so we moved to some other things. We were interested now in knowing whether we can overexpress LSECtin to try and induce some beneficial effect on the cirrhotic liver. So for that, we generated a transgenic model of LSECtin overexpression, and we subject to the same cirrhosis protocol as before with CCL4 for 12 weeks. So first case first, we need to know that LSECtin is overexpressed in our model, and this is what's showing you here in this slide. LSECtin, we compare control mice, the knock-in mice versus the wild type. We'd see a huge amount of LSECtin overexpressed in the liver, and very interesting, there is a persistence of LSECtin in the liver also when the hepatic damage is induced. So this is a very interesting result for us because it allows us to study the overexpression of LSECtin during liver damage, and not only in control situation. So we confirm this again all the time. We have to do all the westerns, all the gene expression analysis because reviewers are very tough, and we have to provide a lot of evidence. It happens to you the same. So next thing we did, so we confirmed the liver damage in our overexpressed LSECtin mouse. Of course, here we have the deciduous red stain. We also have the activation of hepatic stellate cells in the model, but the knock-in model show a reduction both in the expression of pimentin and deciduous red, suggesting a less structural damage in the liver in the case of the knock-in mouse. And at the same time, we also look at cell death with the tandem tumor staining, and we also saw a reduction in DNA fragmentation in the knock-in model of LSECtin overexpression. And in addition to this, from several biomarkers that we used to try and see the function of the liver, so we can see here like ALT was significantly downregulated. The same happened with total bile acids. So somehow we can say that overexpressing LSECtin during cirrhosis is able to contribute, at least can contribute to reduce both structural and functional damage of the liver. What about infiltration in those livers, on immune infiltration in those livers? Well, this is what we're seeing here. We're looking at leukocyte infiltration by CD45. You can see here like there's less white, say, in in the knock-in model versus the wild-type cirrhotic model, and to go a little bit deeper in what kind of leukocytes they were, we saw a reduction in pro-inflammatory monocytes defined as LSECs positive, LSECs negative cells, and at the same time an increase in CD4 T cells compared to the wild-type cirrhotic model. This was kind of a striking result for us. A more CD4 positive T cells, we weren't expecting that, but when we went further in their immune phenotype, we saw that these cells were CD69 positive and PD1 positive, suggesting either exhaustion or regulatory phenotypes. So that led us, more confident with our results. Okay. Going further in characterization of the TH response in our models, so we wanted to know what was the expression of IHM positive, CD4 positive T cells in the liver of our models, where we have like this significant increase in CD4 positive T cells expressing IL-10, meaning flowing tolerogenic towards tolerogenic phenotypes. At the same time, we got the opposite results for TH17. We observe a significant downregulation of the CD4 positive T cells expressing IL-17. So somehow we were reverting this ratio between TH17 and Tregs that is so important in inflammation and restoration of homeostasis. And we tried and confirmed these results by looking again to the transcriptional factors. In this case, you're seeing to FOXP3 for regulatory T cells and ROR gamma T for TH17, and you can see here how the knock-in model has an increase of FOXP3 compared to cirrhosis, to wild type, and a clearly decrease in TH17 compared to the wild type model. So we were rebalancing inflammation toward homeostasis, and this is very cool, right? For us, it's very cool. Okay, so mechanism. We needed to show what was the mechanism for LSECs to induce LSECtin downregulation and how this can retrieve or can change the T cell response. So to do that, we went back to our wild-type model of cirrhosis. Here, we studied IL-17, nothing new. We knew that these guys would have a lot of IL-17 positive T cells in the liver, but we also looked at the ligand LAG3, and these IL-17 positive T cells in the liver of cirrhotic mice also had very significant amount of LAG3, expression of LAG3, right? So this could be a mechanism for explaining this interaction or this modulation of inflammatory response. So we went again back to our cohort of patients and wanted to make sure that this was also present in patients. So we did triple staining for immunofluorescence, CD4 positive, LAG3 positive, ROR gamma T positive, and co-localization of these three different markers was telling us that we have in the liver of cirrhotic patients T cells expressing LAG3 and expressing the phenotype of TH17. So I'm talking now about that the ligand of LSECtin. LSECtin is an LSECs, LAG3 is in T cells, okay? So we can find in patients with cirrhosis these T cells expressing LAG3 and the phenotype of TH17. So again, we always like to keep the translational part alive because otherwise we're disconnected, right? So to work on this and see how this LAG3 could induce LSECtin modulation of TH17 biology, we went back to the lab and then we work on differentiation assays working on lymphocytes obtained from spleen of healthy donors, right? And to look for different things, these are TCR production and cytokines and so on. So how we differentiate cells, we put them with a cocktail of different things that we know that induce immunogenic response. And so the first thing we need to do is to make sure that our differentiation cocktail is including something similar to what we see in nature. So this is what we represent here. Our differentiation cocktail induced IL-17 very potently, so that was cool. And we also saw that they were also expressing LAG3 in a similar way to the primary cells that we were seeing before. So we decided to go further in these cultures, and we, in the following, we added to these differentiated cells LSECtin and LSECtin SC. If we do this, we observe how the expression of IL-17 positive, CD4 positive T cells that had been increased again are downregulated when we add LSECtin to the culture. And not only that, if we measure IL-17 protein in the supernatant of these cultures, we also see this downregulation. So this would suggest, for us, our suggestion was that LSECtin through LAG3 was blocking Th differentiation by inhibiting signals provided by a cocktail, right? But we still need to confirm that, and to do it, we work with an antibody anti-LAG3. And by adding this blocking antigen here, we were able to recover again IL-17 positive T cells in the culture. So this is like very nice in vitro evidence of mechanistic, like a mechanism for LSECtin to provide Th17 blocking in insects, right? Because we know the ligand will block it and everything fits in the same way, right? But still, to go, to be more sure, because this is always difficult to validate, we decided to understand whether, if we need to inhibit T cell differentiation, that is that somehow we have to block TCR activation, right? So for doing that, we just collected the cells, the lymphocytes, but we culture them to induce the expression of LAG3, and when we have LAG3 expressed, we culture them with either anti-CD3, which is an activator of TCR, produce T cell activation, or with IL-6, that is like the trigger for inducing Th17 differentiation, right? And we look at different pathways, and as you can see here, when we had the anti-CD3, for instance, we were able to upregulate many of these pathways, SAP70, ERK1, 2, phosphoic AT. When we added LSECtin, it went back, it went down regulated again, and when we added anti-LAG3, we were able to restore again the expression. And this was not only for anti-CD3, which is the activation of TCR, but also for different intermediaries at the I6 pathway, STAT3 or phosphoic AT. So this is the important drawing here. So we were able to demonstrate here that the inhibition of Th differentiation by LSECtin is through LAG3 by blocking both TCR activation and IL-6 signal. And now we have like the core story to provide a mechanism for LSECtin to induce the modulation of pro-inflammatory response in liver damage. And this is the second graphical abstract. We have demonstrated that LSECs interact with T cells in the liver, and this interaction is established through LSECtin, and this LSECtin is going to interact with its ligand in T cells, which is LAG3. LAG3 represses several intracellular pathways that are going to prevent this TCR activation and IL-6 signaling. Therefore, Th17 differentiation is going to be inhibited. And this is like the envelope for this story. And we now have also demonstrated that we have a very transgenic model of our LSECtin overexpression in cirrhosis or in liver damage that we can use for evaluating future things that we are very interested in. So out of these things, just a few conclusions is that LSECtin plays a fundamental role in modulating the endothelial response to stimuli such as bacterial antigens or their interaction with lymphocytes, which is very important because they promote these tolerogenic responses. LSECtin is repressed during liver damage in patients in animal models. We can reproduce it in vitro and can be positively modulated by anti-inflammatory cytokines, which might be an important tool for endothelial reprogramming in cirrhosis. Maintaining the LSECtin expression during cirrhosis results in less histological damage, less cell death, and less pro-inflammatory local immune environment. With it, just like to keep in mind that reducing Th17 cells is always positive in the context of chronic inflammation. And LSECtin inhibits TCR and IL-6 signaling through LAG3, which is a receptor expressed by lymphocytes in liver damage. So we have a mechanism for providing this attenuation of Th differentiation. So the important question here is whether we can retrieve LSECtin to reduce the hepatic inflammation within chronic liver disease. And this is something that, if we are value, I can tell you if we go for a second project in a few years and I can tell you the results, because we have several options going on. Of course, working with cytokines would be one of them, because we've seen that anti-inflammatory cytokines are able to induce LSECtin very, very nicely. Probably working with them is that we have some technical problems. But we're also testing other options such as epigenetic. I haven't shown you nothing about epigenetics, but we have that unpublished, but we have a lot of data on the epigenetic control of LSECtin through both DNA methylation and histone modifications. So probably going with epigenetic drugs is also relevant to modify the expression of LSECtin. So, but we'll see, can see in the future. Thank you very much.